Hello everyone, my name is Juan. I'll present the poster Development of Plasma-Based Decontamination Treatment for Hydroponic Nutrient Solution. Plasma is the fourth state of matter achieved by adding energy to a gas such that it transitions to a plasma phase. When plasma is used to treat water, we call it plasma-activated water, PAW. When plasma is used to treat nutrient solution, we call it plasma-activated nutrient solution, PANS. This is relevant because controlled environment agriculture, CEA, uses water or nutrient solution to farm vegetables or fruits. If these liquids become contaminated by foodborne pathogens, it can spread throughout the system via its recirculated irrigation. Our study proposes plasma as an alternative sanitation method for the CEA industry. We know from the literature that PAW can successfully inactivate microorganisms in planktonic solutions, but limited research is available for PANS. Our objective was to validate the microbial activation and chemical characteristics of PANS in comparison to PAW, as well as to compare PANS to a common sanitizer in the CEA industry, chlorine. In terms of methods, we had a plasma jet. The feed gas was compressed air. The air plasma treated the surface of a liquid, water in the case of PAW, nutrient solution in the case of PANS, for 5, 10, or 15 minutes, such that PAW 5 indicates water treated by plasma for 5 minutes. The 6 plasma liquids had controls. These were water, chlorine, Cl, nutrient solution, NS, and NS with chlorine. The liquids were tested in their ability to inactivate E. coli DH5 alpha at 2 incubation times, 5 minutes and 10 minutes. Other measurements were the concentration of reactive nitrogen species, RNS, pH, electrical conductivity, EC, and oxidation reduction potential, ORP. When we look at the results, we can see that the controlled water and NS were not effective in decreasing the number of surviving bacteria. Chlorine was able to decrease it below detection level, but NS with chlorine did not achieve the same result. In terms of plasma treatment, shorter plasma treatment times were more effective at longer incubation times of 10 minutes. For longer plasma treatment times, such as PAW 10, PAW 15, PANS 10, and PANS 15, incubation time was not as relevant. In terms of reactive nitrogen species, we measured NO2 and NO3. When we compare the starting point, water, and the plasma treated liquids, we can see that there was an increase in NO2 and an increase in NO3. Same when looking at NS and PANS. An interesting result occurs after plasma treatments of 10 minutes, where it seems that there is a decrease in NO2 and NO3. We hypothesize that NO2 and NO3 are participating in further chemical reactions to produce peroxynitrite, which is another antimicrobial compound. Looking at the pH, ORP, and EC, we can compare the control liquids in the light blue bar to the plasma-treated liquids in the dark blue bar and see that plasma liquids had a lower pH, increase in ORP, and increase in EC. This is due to the plasma treatment generating reactive species and ions, which can then in the liquid participate in oxidative chemical reactions. We conclude that treatments of chlorine, PAW 10, PAW 15, PANS 10, and PANS 15 significantly inactivated E. coli DH5 alpha by more than 6 log CFU per ml, where larger incubation times were more effective in inactivating the microorganism at shorter plasma activation times. For chemical characterization of PAW and PANS, we see similar trends of pH decreasing, ORP increasing, EC increasing, and NO3 and NO2 increasing. PANS is a promising alternative to chemical treatments of nutrient solution to improve the food safety in controlled environment agriculture.